piano. And records.
Welcome everybody to this year's Sir William Paton Memorial Lecture. I'm actually old enough to have known Bill Paton, as everyone called him, and my lasting memory of him was his extreme concern about the use of cannabis. He was aware of so many neuroactive compounds and the lack of any information about their toxicity, and it was a message he spread time and again. I often wonder what he would make of today's pharmacology, where there's an explosion of cannabinoid research with potential therapeutic applications. It would be interesting to know how he would respond to that. But this year's lecture, it's my great pleasure and honor to introduce Gareth Sanger, who I can't think of anyone who could possibly know more about the gut than Gareth. He spent his entire career working on one end of it or the other, and even the gut-brain axis, which of course is an immensely topical subject at the moment. Gareth started in industry with Beecham and then SmithKline Beecham, and amongst his immense achievements are the identification of 5-HT4 receptors and then 5-HT3 receptors as targets for anti-emetics. And of course, subsequently, Gareth was successful in the effort to develop NK1 receptor antagonists as anti-emetics, which was rather more successful than their efforts as antidepressants. That's a sort of personal note. But after industry, Gareth moved into academia, which is a brave move, and was and still is at Queen Mary University of London. And the title of his lecture is extremely long, and I'm sure you can read, so I'm not going to recapitulate that, but I'm looking forward to hearing about the history of anti-emetics, current developments, and future prospects, maybe. Gareth, welcome. Thank you.
Thank you. Well, I apologize for the title. It is rather long, but it is a history lesson, so there's a lot to go through. My first acknowledgement, if you like, is to my friend and colleague, Professor Paul Andrews. He and I wrote this review a few years ago, and I'm going to draw elements from that in at least the first half of this lecture, and the second half brings this up to speed. As a point of interest, we were kind of asked gently to write this review by Professor Tilly Tansey, who gave the very first Bill Paton Lecture both to this society and to the Physiology Society. So that's my small link. I never knew the guy, unfortunately. So let's get on with this quickly. I don't intend to give you a lecture in physiology. You've all vomited at some point in your life. You'll know that it's a complex mechanism, complex reflex mechanism. I'm not going to go into all of this. You know the stomach undergoes retropulsion. You know the diaphragm contraction. The lower esophageal sphincter opens. And the larynx closes to prevent aspiration of vomit contents into your lung. Whoops. Very quickly, the motor nuclei are shown here. The major ones, the nucleus tractus solitarius, it lies in the brainstem and it integrates all of the sensory pathways, the sensory stimuli that make you want to vomit. Don't know how it does that. It integrates and coordinates and sends messages up to these motor nuclei that coordinate that whole physical act of vomiting. The next slide shows you some of the inputs that cause vomiting. And in particular, I'm going to point out the vagal afferents arising from the cardiac regions and the heart that will send afferent information up to the nucleus tractus solitarius to make you vomit, perhaps in response to something nasty you've eaten or to chemotherapeutic drugs. The area postrema, this little red blob there, is meant to represent a nucleus that lies right next to the NTS, but outside the blood-brain barrier. It's been christened chemotherapeutic trigger zone, all sorts of things. But it is responsive to circulating factors, hormones from a variety of different locations, inflammatory mediators, and a note to the pharmaceutical industry, the most common side effect of drugs is nausea and vomiting. It's very common. Abnormal motions and signals to the vestibular nucleus all comes back to the NTS, disgusting sights and smell to the subcortical regions, and eventually lands back in the NTS. And you can see from that, if I can change the slides, there are multiple ways of causing vomiting, multiple conditions, some of which are illustrated here. But there is no single anti-emetic drugs apart from perhaps general anesthesia. It means that we have a whole range of different drugs. Where do they come from? That's the subject of this lecture. And of course, as we see in Ukraine today, there's no better stimulus, I'm sorry to say, for innovation than war itself. This lecture begins with a story in 1914 where they sent a party of, they became known as commandos, out into the sea. They suffered from violent seasickness. And you can see at the bottom, they began to recognize that might be a hazard to the ability of soldiers and sailors to go out and fight. It was the 1930s, 1940s. What was the response? You set up a lot of committees. This was the Flying Personnel Research Committee and the Military Personnel Research Committee, and you'll notice they had some help from clinical pharmacologists and statistics. As is the way with committees, what they came up with was a swing, amongst other innovations, so they could identify those armed personnel who were susceptible to motion sickness and maybe tolerate it out by repeated exposure to that swing. It wasn't long before it became obvious that drug therapy would have to be considered. So it began in the 1940s with minesweeper work. They couldn't do their job in rough seas, so they started doing clinical trials. And look what they did. Trials mustn't interfere with assault training. Simultaneous observation under identical conditions of large numbers of men sailing at the same time in similar craft from the same starting point to the same destination, groups of comparable size, instructed on how to reduce sickness when crowded in small craft, like using vomit bags and eating a biscuit or sweets, and chewing gum given to both groups to reduce dry mouth associated with the ingestion of hyoscine, one of the drugs that was under trial. There you go. It's one of the first structured clinical trials to prove drug efficacy, done in wartime. And the result, you probably know about, hyoscine, the muscarinic acetylcholine receptor antagonist, hyoscine hydrobromide, was the most effective. Today, we know those muscarinic receptors are located in the vestibular system, and we can buy these drugs now over the counter, joyrides and so on, to take out motion sickness. They're easily available. Bringing this up to speed, 40 years later, we figured out the different muscarinic receptor types, and we know that the M3 and the M5 receptors here in the vestibular system, and perhaps in the NTS, are the important receptors. War also saw a lot of research on the phenothiazinic substances. These were identified first in Germany by the textile industry. And you can see a whole lot of dyes, methylene blue, I'm going to come back to that, and the tryptan red identified by Paul Ehrlich as the so-called magic bullet. The story with these substances begins again in wartime, again with the Japanese invasion of the Indonesian island of Java, cut off almost the entire world supply of quinine, used in tropical medicine to prevent malaria. As a result, the armed forces resorted to quinacrine, derived from methylene blue, as I've described. It had activity, but side effects. Look at them, toxic psychosis. It really wasn't very good. USA in particular went back to look at the structure to see if they could modify this, do SAR studies, and find a better compound. They failed. Ron-Polonc, however, the drug company at the time, got interested and saw an opportunity in these compounds to look for antihistaminergic activity. Quite why they thought that, I don't know. They looked at that for the treatment of anti-allergy. The sedative effects of these compounds were later recognized. And they came up with diphenhydramine and promethazine. A little bit of patent busting there by Burroughs Wellcome, came up with cyclazine. And the chemistry template they used, as we'll see later, also introduced an ability to block responses to acetylcholine. You notice I'm not talking receptors. They weren't known in those days. Oops, come on. Diphenhydramine you probably recognize now more as nitol. You can buy that in the chemist. It's an H1 receptor antagonist. That's the data. It's an M2 receptor antagonist. The estimated Ki is about the same. Crosses the blood-brain barrier, and because of the M1 receptor, the muscarinic receptor, because of the H1 receptor antagonism, sends you to sleep and is alleged to give you a good night's sleep. Here's a drug opportunity. You combine diphenhydramine with a compound with 8-chlorotheophylline, in other words, a cup of coffee. Add the two together, and you produce Dramamine, this drug that was designed to have all the benefits of diphenhydramine, but without so much drowsiness, for allergy treatment, and then eventually for the treatment of motion sickness and so on. It was discovered by accident. There was a trial set up at the Johns Hopkins University looking at anti-allergic compounds, and this patient who'd suffered from motion sickness all her life suddenly realized she could travel on the tram cars. The result of that completely accidental discovery launched Dramamine, which again you can buy in the chemist over the counter for the treatment of motion sickness. Here's cyclazine. Cyclazine's interesting. If you talk, I'm not a clinician, but if you talk to the clinicians, particularly in palliative medicine, it's almost like the first line of anti-emetic drug treatment, very, very commonly used. It has the notable achievement of traveling on the Apollo moon missions to prevent space sickness. It's a histamine H1 receptor antagonist, okay, but that's the only real pharmacology I can find on this well-used common drug. I found a pretty rough publication to say it inhibits preganglionic, cholinergic, and vagal nerve activity in animals, so it's probably a muscarinic receptor antagonist, but I don't know. I don't know what else it may or may not do. It's commonly used. I struggled to find phase 3 clinical trial data on this. It's an interesting compound, commonly used. Where's the data? I look at the NICE recommendation for nausea, vomiting, and so on. You notice they're not able to give the indication in which nausea and vomiting was produced. Was it motion sickness? Was it cancer chemotherapy? It's not stated. If you look at the NICE recommendations a bit more, it's incorrect. There's actually no evidence that this drug can take out nausea. And we'll come to that later. So, here's the box diagram again. This is the muscarinic receptors, and the H1 receptors are located here. 40 years later, Ash and Shah proposed the name H1 receptor for this and other work. Histamine receptors that sit again in the vestibular system and perhaps elsewhere, and certainly within the cerebral cortex, can promote sleepiness. Amongst the phenothiazines that Paton worked on, and the definition of the structure here, they were able to identify compounds that had a so-called relaxant effect. In other words, as you'll see, they increased the time required for trained rats to climb a vertical rope for food. And they thought this was great for patients with surgical shock through some of this so-called depressant actions. They identified chlorpromazine in 1951, and a year later it was in clinical practice. In clinical practice for the treatment of cerebral neurosis. A year after that, after its introduction into the clinic, there was a kind of serendipitous moment where Professor Boyd et al. Was visited by Professor Paul, again from France. He had a close relationship with Paton, described unpublished experiments, and described its ability to inhibit apomorphine-induced vomiting in dogs that the drug company had noticed but taken no further. Apomorphine is a complex molecule. It's predominantly a dopamine D2 receptor antagonist, as we recognize today, although it can activate other receptors as well. These investigators found that chlorpromazine could take out vomiting induced by apomorphine in dogs, and that launched a whole new anti-emetic career, if you like, for chlorpromazine. It wasn't until about 30 years later, that chlorpromazine and haloperidol were found to bind to postsynaptic dopamine receptors, work that was in part recognized in the 2000 Nobel Prize. Now, we know that these molecules are actually D2 receptor antagonists, and they work here within the area postrema. That part, if you remember, that part of the brain that's not protected by the blood-brain barrier sits right next to the nucleus tractus solidaris. There's only about one synapse difference between the two. They talk a lot. What's interesting is that today, that the term D2 receptor was introduced, as I say, 10 or 15 years later. We know that activation of D2 receptors causes vomiting. There's a lot of studies on that. What we don't know, though, is if in patients, where does the dopamine come from? Where is there evidence for excessive dopamine to cause vomiting? And that's actually a bit of a concern when you think just how widely used these drugs are and the lack of actually detailed scientific evidence to understand how they work and perhaps their clinical efficacy. There we go, the affinity of chlorpromazine now, to bring you up to date. It's good at D2 receptors, similar to H1 receptors, so it's going to cause sedation with a low selectivity over the muscarinic acetylcholine receptors. So, 1976, we've had all the research in the 40s and 50s. We're left with 4 categories of anti-emetic drugs: the anticholinergics, the antihistamine, derivatives of phenothiazine, and now metoclopramide came along 10 years later. That's a fascinating compound. It originated in France in the Delagrange laboratories under the leadership of Professor Louis Justin Basascon. I apologize for not pronouncing that correctly, is extraordinary. He belonged to the medical committee of the resistance, professor of the clinical faculty of Paris, president of the French Red Cross, co-founder of the Bichat interviews, while running this company and producing drugs like metoclopramide and sulfuride, an extraordinary achievement. What they did within that company was to take cocaine. They wanted to improve on the anesthetic and cardiac antiarrhythmic activity. They substituted the ester group there with an amide group to produce procainamide. They made further substitutions in this benzene ring to make metoclopramide, one of the first substituted benzamides. And of course, needless to say, it had very little local anesthetic activity. What it did instead was to take out apomorphine-induced vomiting in dogs. And it was also discovered to increase gastric emptying and increase transit through the small intestine. They didn't know how that worked. It was subsequently shown, or because of the apomorphine experiments, known to antagonize the functions of dopamine. Later on, we recognized that as the D2 receptor. And that whole mechanism was thought to explain how it stimulated gastric emptying. You'll see that evidence in the literature up until the 1980s, at least it's in there. We now know that it stimulates gastric motility because it activates the 5-HT4 receptor. That wasn't known at the time and is not relevant to the mechanisms of vomiting. We also now know from work done in the 1980s that metoclopramide is a 5-HT3 receptor antagonist at higher doses, and I'll come back to that later. The drug crosses the blood-brain barrier. It causes extrapyramidal side effects like tardive dyskinesia, and so can be contraindicated or has a black box warning, I think, in the US. Its prescription is limited because of that side effect. It prompted Janssen Pharmaceuticals, now part of Johnson & Johnson, to develop their own dopamine D2 receptor antagonist, one that did not cross the blood-brain barrier. They identified domperidone in 1974. Free of the side effects, great anti-emetic, subsequently ran into problems. It was never registered in the USA, and they hit problems with the IV formulation and with the oral formulation interaction with CYP3A4 has limited the use of this compound. So we're at that stage now where we have a bunch of compounds that antagonize at muscarinic receptors, we know what they are now, antagonize at histamine receptors, we know that one now, and antagonize at dopamine D2 receptors, which at that time weren't defined. Then along came an event which the previous speakers in the women's health began to talk about, and again, that's thalidomide. So I apologize for going over some of this again, but it's an important event. Primarily prescribed as a sedative or hypnotic and claimed to cure anxiety, insomnia, gastritis, and tension. And afterwards, it was used against nausea to alleviate morning sickness in pregnant women. As we heard earlier this afternoon, there was not given to pregnant animals. There was absolutely no evidence that this thing may be safe in pregnant women. And indeed, there was actually no preclinical or clinical studies to show that it could inhibit vomiting. Those studies were not actually done until long after withdrawal of this compound. And we see the first evidence of malformed fetuses in 1956. This was an investigator who took some home for his wife. Later on, a growing number of cases forced a withdrawal of thalidomide. What is evident from this compound is that the teratogenicity lies in the S enantiomer. The sedative actions of this molecule lie in the R enantiomer. So it forced drug companies now to consider enantiomers as separate molecules rather than just different forms of the drug. And if you look at new drug registrations now, it's extremely rare to see a racemate because it gives you so many additional studies. And this is just one example of what you can avoid by isolating the active enantiomer. In 1968, in the UK at least, it led to the Medicines Act. So now you had to have a prescription to take this molecule. You couldn't just take it home for the wife. Now you had to have evidence, clinical trials, to inform on the safety and efficacy, animal studies at the very least. And of course, a whole bunch of regulatory studies were introduced. What may seem surprising, though, is that now thalidomide is one of the biggest selling drugs. Biggest selling drugs for a range of different conditions associated with inflammation, immunomodulation, for leprosy, for example, Behçet's disease, Crohn's disease, and so on, skin disorders. It's also used as an anti-emetic. It's still being used, primarily in cancer patients. And these are just some examples of publications that have appeared from different parts of the world, but I have to say most of them are being conducted in China. A lot of those studies, so many in fact, that there have been 2 systematic reviews of all these studies, one published this year, both from Chinese institutions. So it's still commonly used in those countries. Oops. But we've got no idea how it works. Doesn't seem to be related to the ubiquitin mechanisms, the anti-inflammatory, anti-immunomodulatory mechanisms that are important for the other conditions. Here's a study that was done, I think, in 1998, a study which needs to be repeated. There are some faults in this study, but I think you get the message. It doesn't have affinity or binding efficacy, binding ability to any of these ligands. That's what they claimed. It includes all of the receptors that I've talked about before that are involved in mechanisms of vomiting. Doesn't touch them. We looked at that recently, and we've just, I'd hoped it be accepted by now. It's still being thought about by the referees, but we've looked at that and we've come up with a range of different actions that might explain how this compound could be anti-emetic, including a reduction in the secretion of growth differentiation factor 15, or GDF15. And incidentally, going back to the women's health discussions today, GDF15 is extremely high in patients with hyperemesis gravidarum. It causes vomiting. So I'd love it to work that way, in which case we have perhaps the beginning of chemistry to antagonize at that receptor. Then along came cisplatin. Cisplatin's a compound prescribed and used in its different forms for the treatment of cancer. It belongs to what is called the highly emetogenic anti-cancer drugs. You can see some have got low incidence, some are moderate, and some are severe. Cisplatin is particularly rough. Here's an example. About an hour and a half after one administration, the patient will go into severe paroxysms of vomiting and nausea, dies down a little bit, this is only a cartoon, and then comes back on day 2, 3, 4, and so on. This is a single administration. The patient then comes back for repeat doses and so on and so on. In 1985, 20% refused treatments. None of the drugs I've talked about before worked. There were tears, there were vertebral collapse, there was metabolic acidosis, there was anorexia, there was stress, there was incapacitation, people lost jobs. The intensity of the vomiting induced by cisplatin was reduced a little bit by people who smoked marijuana, and that led to the use of the delta-9 tetrahydrocannabinoids to take vomiting out a little bit. They're not fantastic against cisplatin. They do have some effects. They're great at appetite promotion, but of course limited by side effects. They work here in the dorsal vagal complex within these brainstem motor nuclei. Then along came Richard Gralla. He's an oncologist in the US who clearly had zero training in pharmacology. He looked at the phenothiazines and the cannabinoids and all of the drugs that I've talked about, and they weren't working. He figured out that in the US, since metoclopramide was a relatively new drug, although widely used in Europe, and since the dose wasn't well established, he would escalate the dose to maximize the chances of success. As I say, he had no pharmacology, I assume. And this is a quote from a witness statement. I looked at the world's suicide literature, and it looked as though it was impossible to kill yourself with this drug, so that sounded good. he published what became a landmark study in 1981, showing that high doses of metoclopramide could reduce substantially this vomiting induced by cisplatin. That was a major study. It was assumed metoclopramide worked through D2 receptor antagonists, so there was a whole raft of other studies to say, well, okay, if that worked by bumping the dose up, maybe we need to bump the dose up of other D2 receptor antagonists, perhaps to get more in the brain or so on. And I remember when I worked in the pharmaceutical industry, that was the prevalent theory at the time. You can see here, none of them worked. Some of them caused additional side effects, complete disaster. The efficacy was not replicated. When that was published, at the same time, John Fozzard, then working at Merrill Dow, was promoting or developing and redefining the ability of relatively high concentrations and high doses of metoclopramide to antagonize what were then known as the 5-HT-M, became 5-HT3 receptor. We put those two pieces of information together, and together, while I was at Beecham, published this study to show that you could take out cisplatin-induced vomiting in ferrets by selective 5-HT-M or 5-HT3 receptor antagonism. What we did was to use the world's first selective antagonist at this receptor, MDL72222, developed by John Fozzard and his team. So what I did was to write to him. I worked at Beecham, he worked at Merrill Dow. Today, a whole army of lawyers would be on my back still if I were to do such a thing. But John was kind enough to send me a copy of his latest manuscript that described the molecule and also a sample of the compound. And it was that that led to this study here. I don't think you could do that today. The 5-HT3 receptor, to cut a long story short, works down here. It's about 90% of the body's 5-HT is contained within the endocrine cells lining the mucosa of the gastric antrum and duodenum. When it's released, it sensitizes and stimulates the vagal afferents to eventually promote vomiting. It is effective against the acute chemotherapy. That is the first 24 hours. Remember I showed you that sort of biphasic curve. There's some effects later on, but not good enough. So it became regular practice to combine a 5-HT3 receptor antagonist with dexamethasone to take away the consequences of cell damage caused by cytotoxic therapy. It substantially reduced, it changed medical practice, and it substantially reduced the cost. This is interesting, because the hospitals no longer had to wash bed linen, wash clothes, provide vomiting buckets, no longer had to admit a patient into hospital. You can now do these treatments in outpatients. So here's a rare example of a drug that actually reduced medical costs. And a whole load of compounds were developed and are on the market as drugs. I'm going to come back to paloncitron, one of these that was introduced relatively recently, in a few slides later. Of course, rather than just block the action of one substance, why not take out the majority of the vagal nerve activity by antagonizing the ability of substance P to activate the NTS and also which was also involved in some of the motor nuclear activity, by developing an NK1 receptor antagonist. We have one now, Prepident, and also the IV formulation. It blocks the action of substance P and is now a highly effective anti-emetic. This is an old slide, but it hasn't changed. 3 studies, 1, 2, 3. They looked at the overall vomiting burden in patients in the dark green that received a Prepident or did not, just received a 5-HT3 antagonist plus dexamethasone. The acute and the delayed. And you can see that it substantially improved the delayed vomiting in these patients compared to those who received just 5-HT3 plus dexamethasone alone. So now we have recommendations for severe forms of vomiting. You have to give a 5-HT3 receptor antagonist plus dexamethasone plus NK1 receptor antagonist. And for moderately severe, you can leave out the NK1. Here's palontotron. I promised you, this is different. This is the only 5-HT3 receptor antagonist that's licensed for use against acute and that delayed vomiting. And the reason is it's thought not to actually bind to the 5-HT3 receptor in the conventional competitive manner, but to bind allosterically. That leads to receptor internalization, and somehow the internalized complex desensitizes the NK1 signaling pathway, so there is believed to be some intracellular crosstalk, and hence you get a lot more bang for the buck, as it were. That's why this drug is registered for both. I think there's a lot more work to be done there, but that's where we are at the moment. I said it's an unfinished tale. You'd think its job was done. It's an unfinished tale because of this, nausea. I don't need to define that. You've probably all experienced it. It's nasty, unremitting. You look at the clinical trials, and the latest I've got here is 2015, but I'll show you one in 2019 at the moment. Read that. 31% of cancer patients didn't get a complete response. Nausea worse controlled than vomiting. Late phase worse controlled. So we begin to get titles like nausea, the neglected symptom. And it's not just cancer patients. It's also a range of other conditions I'll show you later. What's it like? These are some quotations taken from a 2019 publication. And I'll draw your attention to that. These are patient responses. It's kind of like there's a word in the language that's missing, because nausea is, I feel like I'm going to be sick. Then there's this nausea. It seems like there's a big blank space in between. There's a missing word. Just to bring home what the patients feel. This is 3 years ago. The response so far has been the introduction of olanzapine, which often people will give now with a 5-HT3, with an NK1, plus dexamethasone. And you look at olanzapine. It's an antipsychotic. Look at what it does. D1, D2, D3, D4, 5-HT2A, 2C, 3, 5-HT6, the alpha-1 adreno, H1, M1 receptors. Some improvement in nausea. But I draw your attention to that H1 receptor and the increased sedation with olanzapine. So I would want to know whether it was the sedation that was helping those patients or whether it genuinely had any anti-nausea activity. So we've got anti-emetic drugs, which by definition, from its historical derivation of the word block vomiting, a lot of people assume it takes nausea out. But I'm beginning to tell you that nausea is not well treated by these drugs, and I'll show you other examples. So we do need to introduce the term anti-nauseant drugs, a new class. The answer is no. There's a paper published in the British Journal of Pharmacology last month by Seifert and Steve Alexander, who argued that we should replace the terms which originally described the therapeutic orientation of a drug class. Anticholinergic, for example. What does that mean? Muscarinic, nicotinic, so on. What does it mean? With more accurate descriptions of the molecular target, like muscarinic acetylcholine receptor antagonist. And they gave a lot of other examples, antispasmolytic for the gut. What does that mean? There aren't any spasms, and yet this is commonly used. My colleague Paul Andrews and I love that article, but we also found a few objections. So I'm delighted this letter in response was just been accepted, so it should appear soon. And its proposal to consider the gut. It was interesting that in the original paper, conditions associated with gastrointestinal disease were kind of tucked under other, a class down there. We kind of didn't like that too much, but our main problem with this wonderful paper was that they proposed to abolish the term anti-emetics and replace those, as I've described, with the molecular targets of the different drugs. And they argued that this would now be, these molecular targets would now be prescribed for the treatment of nausea. So suddenly a bright idea suddenly got confused with this idea of anti-emetics treat nausea. They don't. And a complete mixture. And that's why we wrote the letter, complementing the paper to begin with. So, nausea, what's a good model? You can give a drug, but there's no escape. You can induce motion sickness, but the kit is probably very expensive. Or you can make people nauseous by virtual reality. put them in a driverless car, for example, will usually do it. But more often now, it's one of these little virtual reality machines. Here's an example of a study. Here's a volunteer, has an MRI scan, lying prone, looking at visually alternating black and white stripes going around in a circular motion until they progressively feel nauseous. And nausea was defined here as epigastric discomfort. There are many definitions of nausea, which makes clinical trial an issue. Candidate brain regions for processing nausea include those associated with interoception, and other studies may play a role in emotional and the emotional disgust related to nausea. These are the brain areas that light up. For those of you who may not be aware, let's consider the word interoception. This is the sense of knowing what's going on in your body. This is the sense of, for example, coming up here and feeling butterflies in your stomach and maybe your heart racing a little bit. Stuff that you wouldn't normally feel, stomach issues. Interoception became an issue here where, with some colleagues, some fantastic colleagues in Spain, did a systematic review on all of the studies that had looked at brain imaging in patients and volunteers experienced nausea. And there weren't many. I think they had about 17 in the end. But they highlighted these areas here that you'll see in color and concluded that during nausea, there's a sustained activation of the interoceptive, we're back to that word, limbic, somatosensitive, and cognitive brain areas, cognitive, of course. Consistent with nausea as a multi-dimensional perceptual state, irrespective of the stimulus, and of course limitations, the poor number of studies, the poor definition of nausea, and the different techniques. So limited and just been accepted for publication. So this word interoception made us look back at the stomach, and the stomach, as you may know, is composed mostly of smooth muscle, but with a myenteric plexus of neurons within that helps to control some of the motility. Contained within are also the interstitial cells of Cajal around the myenteric plexus and deep in the muscle layer. These are cells that spontaneously depolarize, and they're connected by gap junctions in a syncytia. When they fire, up here, there's a sort of pacemaker area here, they send a wave of slow wave electrical activity around the stomach and down to the pylorus. The logic being that that keeps promoting gastric emptying in an anal direction. If you can't digest your food, you're dead. So this thing is a continual push towards the right direction. Supplemented, of course, by neurons and hormones that act on that baseline. So we can measure electrical activity indirectly. We can stick electrodes on the abdomen and do an EGG, an electrogastrogram. Very common. Criticized, but very common. And you see here a volunteer, 3 cycles a minute activity, this electrical rhythm of activity. Now stick them in an optokinetic drum, make them nauseous, and you see that goes wild. The frequency goes up, there's tachygastria, there's dysrhythmia, completely changed. And we can do that, others have done this in a lot of different conditions. You see them all here. This is a commonly observed event during nausea. I'm more interested in the gut, of course, gastroparesis, functional dyspepsia, chronic unexplained nausea and vomiting. These are conditions where, apart from metoclopramide in most major countries, there are no registered drug treatments. These patients get a mixture of anti-emetic, gastric prokinetics, almost at a whim. There is no registered treatment. So they're important. Here's a study done by Greg O'Grady. He's a surgeon in New Zealand, interested in electronics. And what he did was to design a flexible printed circuit board that when he went into the patient, he could put on that and measure the electrical activity in the stomach under anesthesia. That's a weakness. Ignore these things, they relate to the videos he made, but look at those arrows. 3 cycles a minute, consistent with the EGG, nice uniform direction of electrical activity. Then he did it in a patient with diabetic gastroparesis, and look at those arrows. They're kind of all over the place. This was a patient which had tachygastria in a relatively stable manner. He found irregular tachygastric and random ectopic events. So this patient had really badly and dysregular electrical activity within the stomach, and they suffered from nausea. That's a major symptom of gastroparesis. So, what causes it? There is some evidence of damaged interstitial cells of Gahal. That could be one example. I'm getting near the end here. But neuronal, hormonal, and or cytokine activity acting on the stomach could also damage. And here's an experiment that my colleague Raj Makwana is going to describe to you in the next session that follows this, so I'm going to be fairly brief. This is a human stomach, spontaneous myogenic contractions, that's muscle. The contractions occurred about 3 in a minute. They're not affected by tetrodotoxin. They're reduced by the calcium-activated chloride channel blocker MONA, which is fairly specific for the ICCs relative to smooth muscle. So it looks like this is an ICC-generated event. We add vasopressin. Vasopressin is released during nausea and vomiting. It causes nausea and vomiting, and we see some kind of increase. We then, in a separate experiment, add adrenaline, released during stress and nauseogenic conditions, gain a kind of interest. Add the two together, look at that. Raj is going to describe that data. He's characterized those contractions. We've quantified the rate of contraction, the rate of decline, the area under these different components. And to cut a long story short, that kind of mismatch of color is the control plus vasopressin or adrenaline given individually. But look at the purple line when they're given together. A massive increase in contraction amplitude, contraction frequency, the rate of contraction development. And we hope to go on to use this model now to look at the effects of other nauseogenic stimuli to begin to investigate how they may be provoking this whole condition by acting on the stomach and on the pacemaker activity of the stomach. So I've whistled through that fast, talk to Raj later. There's the title. I got it wrong. I shouldn't have said anti-emetic drugs. We now should modernize that and say drugs preventing vomiting and or nausea. I think they're different. It's an epic marked by war and trial design. I mentioned the motion sickness drugs. It's marked by tragedy and the subsequent improvement in regulation. That's the thalidomide story we heard about earlier. Many drugs were identified before the receptors were identified. That kind of indirectly helps pharmacology because there was a response to measure. Chemotherapy, the 5-HT3 receptor, dexamethasone and NQ1 revolutionized clinical care and continues to do so. And of course, it's an unfinished tale. Nausea is still out there. It's a major symptom of many different clinical conditions, poorly treated. I said that it completely changed the pharmaceutical industry. You can't read that, I'm sorry. But during the lifetime of the research that I've talked about here, 32 companies are now 8. Sanofi, GSK, Novartis, Roche, Astellas, Pfizer, Johnson & Johnson, and Merck. The rest have all gone. So I hope this has been a talk that meets with Bill Paton's approval. I hope there's some history in here, which I hope he would have loved. It remains for me to say one last thing, and that is to kind of dedicate this talk to my friend and colleague Wes Minor, shown here on your right. He conducted and pioneered all of these studies that we did using the ferrets to show how these drugs vomiting, and he changed the design in such an effective way. He could stop vomiting in its tracks, he could do all sorts of things. And I'm sorry to report he died of cancer last year. So that's the end of my presentation. I'm sorry if it was quick, but I had very little time. Thank you very much.
And thank you for a most interesting lecture. That was really fascinating and instructive. Unfortunately, we've run out of time, so we're unable to offer open questions, but I'm sure Gareth will be pleased to answer questions informally over refreshments or over the dinner. Meanwhile, I just have one final role to play, which is to, Gareth, don't go away, to present you with your certificate.
Okay, thank you very much.